Hello everyone, welcome to the Knowledge Hub by Sri Chaitanya. This is Sayyid Danish Anjum, Senior Zoology Faculty, and today I'll be going to take some basic concepts of animal classification. So let's start our very first session of animal classification. In this session, we are going to discuss some of the basic concepts of animal classification, which are some of the basic parameters upon which the classification has been made, and the next part will be classification of animals. That means the proper phylums into which all the animals are categorized. We will start from this topic, right? So starting with this session, what questions or what type of questions we can expect from this chapter is identification of phylum and its respective characteristics. Every phylum is having some special characteristics, its own qualities, its own characteristic cells. So in examination, NEET examination, the questions which are used to ask is to match the characteristics with its phylum or the examples of the phylum. The second is the match the following type, which you have seen in this examination also, and scientific names and common names, which are very common and they come regularly in match the following category only. The fourth is symmetry and coelom. That means what phylum or the organism in that particular phylum is having what kind of symmetry and what kind of coelom along with the germ layer organization, whether it is diploblastic, triploblastic, like this, and along with common characters among different phyla. So if we see what are the general characteristics of Animalia, that means if we discuss what are the qualities upon which we can define an animal. So in general biology, we call metazoans the true eukaryotic multicellular animals, and apart from that, they are having other characters. That means the sole multicellular eukaryotic character is not the only criteria upon which we can classify the animals, because there are other multicellular eukaryotic organisms also. So the other important character is the heterotroph. That means they are having a very specific mode of nutrition, heterotrophic, but as we all know that in heterotrophic mode of nutrition also we are having saprophytic, parasitic and holozoic mode of nutrition. So in this category, heterotroph, the animals are having holozoic mode of nutrition. Now if we talk about the holozoic mode of nutrition, we can define it as the engulfment of the solid raw food particle, either whole or either in what we can call it as partly, either solid or in some cases liquid also. So if an organism is ingesting or swallowing the food particle, afterwards digesting it in the digestive cavity, and absorption of the digested food particle and the assimilation of the digested food particle so that it could reach out to every cell of the body or organism. So this is the pathway or the steps of the holozoic mode of nutrition. So if I have to define, I can divide in like ingestion, right, digestion, absorption, and at last assimilation. So these are the steps of holozoic mode of nutrition, and you will also study in biological classification that one of the key parameter for the classification is the mode of nutrition. So if we take the other characteristic which is very peculiar to the animal classification, it is this one, nervous system. Now if we talk about the invertebrates, the lower category of organism or lower category of animals, we are not having a very proper defined nervous system, although you will find examples that they are having nerve net kind of structures. But in usual category, if we look at the higher level of organisms in animal classification, we will find a proper nervous system along with chemical control system. Chemical control means endocrine system, hormones, and nervous system, they act in a coordinated manner and they make the organism conscious or aware of its surrounding. Okay. So these two are one of the key characteristics while we define animals. The other is the animal's cell usually lacks the cell wall. Now, the question arises, why the animal cell lacks the cell wall? It is because the endoskeletal support is played by the actually bones which are of cartilaginous or bony in nature. So, the cell usually do not require cell wall for rigidity because the organism body support is provided by the endoskeletal support, which could be either cartilaginous or it could be bony in nature. Correct? The other is the general feature, it is motility or locomotion. Now we have known that every movement is not a locomotion, but every locomotion is a type of movement. So in general, locomotion is the movement in which organism uses its locomotory structure or locomotory organ, and it changes its place from one place to another, or it changes its location. Now the other is, all the animals are having mostly sexual reproduction, and it is also a source of variation, but it is not the sole characteristic. Animals, particularly the lower invertebrates, are having asexual reproduction also along with the sexual reproduction. So these are the general characteristics to define animals, right? So if we look further, what are the different parameters for categorizing all the animals in different phylums? What are the basic parameters? The first parameter is the level of organization. Now we will discuss each and every point one by one. So we will have a look at all the parameters first. The first is the level of organization, starting with the very first basic cellular level, afterwards tissue level, organ level, and finally the organ system level of organization. The most complex is the organ system, and the most basic and the simplest is the cellular level of organization. Followed by symmetry. Either the organism is asymmetrical in nature or it is symmetry. If it is having some kind of symmetry, then we further classify whether it is having bilateral symmetry or it is having radial symmetry. The third is the germ layer organization. That is, if it is having germ layer, how many germ layers are there? If it is having two germ layer, that is ectoderm and endoderm only, then we will call it as diploblastic. Or if it is having all the three germ layers, that is ectoderm, endoderm and mesoderm, we will call it as triploblastic organization. So this has to deal with the germ layer organization. That means after the formation of zygote, the cells form the germ layer, basic primary germ layers, and from all the germ layers, that is ectoderm, mesoderm, endoderm, all the other type of cells, all the other tissues and organs are forming, right? Next is coelom. Now coelom is a very interesting parameter here. We will discuss in detail about how the coelom is forming. Apart from the general coelomates, we will discuss enterocoelom as well as schizocoelom also. So coelom is basically it is defined as that particular body cavity which is enclosed by mesodermal layer. So this is the proper definition of coelom. So the first important point is the organism must be having body cavity. Now every body cavity cannot be termed as coelom, because the other condition is that that particular body cavity must be enclosed by mesodermal derived layer or mesoderm germ layer. Fine. So we will discuss this coelom along with the examples and diagrams. The other is the fate of blastopore. Now what is blastopore? During embryonic development, we will discuss it before coelom only, but just to have a little indication, during embryonic development, when the germ layers form and the invagination process occur, the gastrulation process occur, during that process, the blastopore is this opening. This is the archenteron cavity, this is the ectoderm germ layer, this is the endoderm, and mesoderm depends upon schizocoelom or enterocoelom. We will discuss there only. So this particular opening, as you can see my marker, this particular opening is called as blastopore. Now in some animals, this particular blastopore, if it develops into mouth first and the opposite point into ns second, then such kind of animals are called as protostomiates. Or in vice versa manner, if this blastopore opens, if this blastopore develops into ns first and the opposite pore or opposite side develops into mouth first, then such kind of organism are called as deuterostomiates. So we will discuss when we will discuss the fate of blastopore. The other is the segmentation. Now segmentation is a very interesting phenomena, and it is also the most confusing one to the students, because true segmentation, pseudo segmentation and metamerism are some of the terms which are usually overlapping. So we will discuss segmentation also, that if the organism is segmented, then if it is a true segmentation, or to identify whether it is a pseudo segmentation or whether it is having a metamerism, that we will discuss along with the examples. So the next and the final is the notochord. Now students, notochord is basically a structure which is developing at the dorsal side below the nerve cord and above the alimentary canal. It is of mesodermal origin and its main function is to provide endoskeletal support. So we will discuss when at the end we will discuss about when we will start the chordate, we will discuss about the notochord formation in depth. Okay. So in summary, if we define the level of organization, metazoans means all the eukaryotic multicellular organism. So basically all the animals are divided into two categories. The first, the primitive basic category is the parazoa, which is having the cellular level of organization, and it is the only one which is having the cellular level of organization. Rest of all the animals are categorized into eumetazoa. Eu means true, metazoa means animal. So eumetazoa are having all the other level of organization, as you can see, tissue level, organ level, as well as organ system level of organization. Here are some of the examples also we can see, and we will see along with other more examples as we will proceed. Now, starting with the very first basic parameter, level of organization. The very first simple level of organization is the cellular level of organization. Now, what are the characteristics of cellular level of organization? The first point is the cells are loosely aggregate. That means although they are multicellular, there are many cells, they are in a loose aggregation manner. What do you mean by loose aggregation? That means here we are having 2 points, that all the cells more or less are behaving in an independent manner. That means a very little coordination will be present among them, but still if we talk about the cellular level of organization, that means the cells, all the individual cells which are present in the organism's body are behaving more or less in an independent manner. The first point is this. And the second point is they are having very little coordination among themselves. But you must remember, there will be a little coordination present, but not to that extent, so we can call them as a tissue or further advanced organ. So a little coordination will be there, and due to that a little division of labor will also be there. So present. So these are the some basic points of the cellular level of organization. So you remember the very first line, that cells will behave in a very little coordinated manner. They will be having a little division of labor among themselves, but more or less they will behave independently, right? So if we see that how the level of organization starts, it starts from the atom to the molecules, macromolecules, organelles are forming, organelles assemble, a complete cell is forming, then cells are coordinating and cells are aggregating in a similar structural and similar functional manner, then tissue is forming, and the tissue is aggregating, it is forming organ, and when organs are forming a system together in a coordinated manner, organ system level is forming, and from organ system level the organism is rising. So this is how the level goes up, starting from the cellular level to the organ system level of organization. Now let us look further. Starting with the cellular level, as I mentioned you that the very first basic example of this is the phylum Porifera. So here you can see the Porifera. This we will discuss further as we will start the Porifera. This is the water canal system movement. You will see how the water input enters into the body of the poriferans and it is exported out through the osculum. So what are the basic parameters, basic points? Cells are arranged as loose cell aggregates. Already discussed. Some division of labor along with some little coordination will be present among the cells. So examples are poriferans. Only single phylum is the example, right? We call them as sponges, we call them as pore bearer, etc. Now starting and coming back to the next level is the tissue level of organization. Now students, in the lower classes, 9th and 10th, we have studied about the tissues. The definition of tissues, what it says, that generally when you define tissues, you define that cells which are aggregating, which are having similar structure, which are having similar function, and along with similar origin. Those aggregation of cells is termed as tissue. So similar origin, right? Similar origin, similar structure, and function. So if they are behaving in such a aggregated manner, in such a coordinated manner, that means in comparison to the cellular level of organization, they are having a very good coordination among themselves, a very good division of labor among themselves. So here in this tissue level of organization, all the cells in a particular type of tissue will be having a very good coordination as well as high division of labor among themselves. Correct? So arrangement of cells becomes complex, definitely, as compared to the cellular level of organization. Tissue level is advanced. Second is cells performing same function. I must remove this. You can write it down later on. I'll dictate it further. Cells performing same function aggregates into tissue and also same or similar structure and along with similar origin. That you must keep in mind, right? Okay. Examples are, the basic examples of tissue level organization are two phylums, phylum Coelenterata or Cnidaria and Ctenophora. Here we can see the example of Cnidaria. This is sea anemone, Adamsia, and this is the jellyfish, which is also showing the characteristic of phylum Ctenophora, that is bioluminescence. We will discuss it further in the upcoming phylum Ctenophora, right? So this is the tissue level organization. Now the next advance is the organ level of organization. So when the tissues are grouped in such a manner that they are having coordination among themselves, division of labor further increases, then advanced level of organization, this advanced level of organization is termed as tissue level of, organ level of organization. As we can see that the example is platyhelminthes, which we usually also call them as flatworms. Here we can see organs is composed of multiple tissues and all the tissues are behaving in a coordinated manner and division of labor is also present among themselves. As a result, a highly specialized structure is forming, which we are calling it as organ. This is nothing but the example of platyhelminthes tapeworm. Okay, so we will move to the organ system level of organization. Not to mention further, when organ will assemble, they behave in a coordinated manner, the division of labor further increases, efficiency of the work function further increases. As a result, organ system level of organization is the most advanced character. So group of organs associate to form an organ system, and each organ system is concerned with a special physiological function, right? For example, if we talk about the digestive system, there are various organs like the gut itself, liver, pancreas. There are various organs in the digestive system which act in a coordinated manner, and all of them are having a specific physiological function of digestion, right? Similarly, the excretory system is there. Similarly, circulatory system is there. So in every system, multiple organs are coordinating with each other to form a specific system and to carry out a specific physiological function. Okay. Example we can see from Eschelminthes, right from Eschelminthes or Nemathelminthes to chordates. We are having organ system level of organization. Here we can see. Okay. So the next is digestive system. Now digestive system, if we talk about in general, in some organism you will see digestive system is absent also. In the lower poriferans, the digestion is restricted to individual cell. But if we talk about the digestive system, then usually it is categorized into two parts, two distinct parts. The first is the incomplete digestive system. When we talk about the incomplete digestive system, here you can see the incomplete gut. It is having a single opening, mouth. Wait a minute. Like this. Okay? It is only having a single opening. The other opening is closed. So if we look at this kind of digestive system, we usually call it as blind sac body plan. What we call it as? Blind sac body plan. So I am writing it as blind sac body plan. That means the other end is closed and there is only one single opening which is used for both ingestion as well as egestion, right? Examples you can see Cnidaria, Platyhelminthes. In Platyhelminthes you will find that in example like Taenia, the digestive system is absent. So apart from that, the organisms are having incomplete digestive system or blind sac body plan. Now looking at the other advanced and more coordinated system is the complete gut system. This type of system we call it as tube within tube body plan. Okay. Tube within tube body plan. The first parameter is that it is having two openings, mouth as well as anus. One opening for ingestion, the other opening for egestion or defecation, right? So we can consider it as whole body of the organism. Now you look at it. This whole body of the organism can be considered as a single tube, and in that particular tube, a separate tube of digestive tract is present starting from mouth to anus. So we can consider it as tube within tube body plan. Okay, so this type of complete digestive system is also known as tube within tube body plan with the two separate openings. Fine? Okay, so some organisms are having incomplete, some are having complete. Next is the circulatory system. Now if we talk about the circulatory system, as we have seen in the digestive system, there are two types, incomplete and complete. Similarly, if we look at the circulatory system, they are also categorized or divided into two parts, that is open type and closed type. Now open type and closed type circulatory system are concerned with the body cavity. One is concerned with the body cavity, that is open type circulatory system, and the second one is having a closed circulatory system, that means it is having a closed network of blood vessels. Now we look at the open circulatory system. So what are the general characteristics of an open circulatory system? What are its requirements? First of all, we need an open body cavity. Now the body cavity is called as hemocoel, and open-ended large blood vessels are there. That means blood vessels will be there, but you can call them as rudimentary developed or very open large blood vessels developed. So we are not having a very proper network of blood vessels. Proper close network will not be present, but a rudimentary developed and open large blood vessels will be there. So cells and tissues are directly bathed in it. That means all the organs, all the tissue will be in complete contact with the circulatory fluid. Now in here, open circulatory system, that fluid is called as hemolymph, which is circulating in the body cavity, known as hemocoel. So in hemocoel, the fluid which is circulating is called as hemolymph. Now there is a slight distinction between hemolymph and blood. What is the difference? That if we separate blood and the tissue fluid separately, that means blood is separated with the tissue fluid, from the tissue fluid, then blood is flowing in the blood vessels, and tissue fluid is basically that part which is the filtered plasma from the blood capillaries. But in hemolymph, the blood is mixed with the tissue fluid. There is no separate distinction. So we call it as hemolymph. And hemolymph circulates in the cavity which is called as hemocoel. Fine. I will give you the example, the schematic diagram, and explain it further. So fluid is hemolymph. All the cells, all the blood cells are also present in this hemolymph only. Example, we can see arthropoda and mollusca, right? Here we can see the schematic diagram. There will be a heart. Now heart could be somewhat tubular heart, or it could be another kind of heart. And here we can see the blood vessels are there, but they are not completely developed. And this is the cavity, this is the enclosed body cavity in which the hemolymph is circulating. So from heart it is coming into the sinuses. I will explain the sinus also. And from sinuses it is going back to the heart, and the circulation continues like this only. So we will see further how does it happens. So the best example to explain the open circulatory system which I take is of the cockroach. You will study in the cockroach also, its open circulatory system. So if you see the cockroach, this is particular the heart of the cockroach. Now heart of the cockroach is a tubular 13 chambered heart, and here we can see that, let me tell you in this diagram, the ostium, this is the heart, and ostium are the pores through which hemolymph enters into the heart, and heart pumps into the major artery, and through major artery it is pumping into the sinus cavity, and through sinus cavity it is going back into the heart via ostium. Let me explain it to you in this particular diagram. Let us suppose the other eye I cannot draw. You can consider that it is having two eyes. So let us consider that this is the whole body cavity. So the heart of the cockroach is at its dorsal side. It is tubular heart, 13 chambered heart with anterior aorta like this, and its whole body cavity, that means whole hemocoel, we call the body cavity as hemocoel. It is also a large sinus cavity, but in actual, if we divide the body cavity into separate compartments, then each particular compartment can be called as a sinus cavity. Basically sinus word is used, sinus cavity is used for that cavity in which the blood or hemolymph is circulating, right? So in cockroach, the body cavity is divided into three parts: pericardial, perivisceral, perineural. You will study later on also. So what happens in general, the blood enters into the 13 chambered tubular heart via ostium, and each chamber pumps the blood into the anterior ones ahead of it, and in this manner through the anterior aorta the blood enters into different respective sinus cavity, head sinus cavity, perineural sinus cavity. So these are the membranes through which the hemolymph will enter into distinct chambers, distinct sinus cavity, and ultimately this circulation will be completed when the hemolymph will enter into heart again via ostium. So this is the basic idea of how does the open circulatory system works. Okay, so looking at the closed circulatory system, it is not of special, it is of special significance. Definitely, we are having the closed circulatory system, some lower invertebrates also, for example, annelids. So, what are the different characteristics of or different requirements of the closed circulatory system? The first one is the blood itself, which is a circulating fluid, and the blood is circulated through a series of vessels of varying diameter. Varying diameter means artery is having a diameter, vein is having a diameter, capillary is also having a diameter, but the diameter varies from artery, veins and capillary. We will discuss each one by one when we will discuss the closed circulatory system further in detail. Okay, so we are also having a very close network of blood vessels in which the circulating fluid, blood is flowing. Okay, the second is exchange of material between the blood and the tissue is occurring by the interstitial fluid, or we can call it as tissue fluid. Okay, again heart will be there. It is a basic pumping organ which will carry out the circulation in the blood vessels. Fine. So blood circulation is precisely regulated, and examples are Annelida, the lower invertebrates and vertebrata. So here we can see, this is the Annelida diagram, earthworm, pheretima, and heart is pumping into blood vessels. These are the capillaries, small capillaries through which the tissue fluid or interstitial fluid is filtering, and it is coming in contact with the surrounding tissue, surrounding cells, surrounding organs, through which the exchange is occurring, and afterwards the rest of the blood is pumping back to the heart via veins. So through arteries, capillary, vein, heart. So this is the closed circulatory system network. Okay. Now moving further, there is a small, you can call it as a quiz. We give you, like, you can take a minute or two to read and guess the answer. In the next slide, we will display the answers. All members of Animalia are, cells are arranged as loose aggregates in which level of organization, and so on. These are the very simple kind of fill in the blanks. I hope you have read out. So let's see what are the answers. So these are the answers, right? So these are the some of the basic general questions. The depth of the questions can be further increased up to assertion, reason, match the following. So up to this moment, I hope I have made myself clear about all of the points. Now let's move further. Talking about the another important basic parameter of classification is the symmetry. Now to understand the symmetry, we require some imagination. Alright. So if you want to know whether an organism is symmetrical or asymmetrical, just by looking itself you can say, but to be precise in terms of the language of biology, you need to have a proper method. Now what does this proper method say? For example, if you are having a body, for example, I will be taking a minute. Sir, I need this bottle, plastic bottle. Yes. Thank you. So you can consider this as an organism body. I am taking this as an example. Now, to know whether the organism is symmetrical or asymmetrical, it must be having identical half upon division. Now that division must be imaginary only. So let us suppose this is an organism, this is the body. Another important point, symmetry is the characteristic of morphological appearance. It has nothing to do with the anatomical, because if suppose you divide me into two identical half, my morphological body will be into identical left and right, but anatomically it is not possible. So it is a morphological feature. So if you want to know whether this organism is symmetric or not, what you have to do is to imagine an imaginary axis which is passing through the center of the body. Now you have to imagine a center of the body in between, in the inside of the organism. So when an imaginary line is passing through the center of the axis, center of the body like this or like this, and when you take an imaginary plane and you divide the organism body along the imaginary axis, and if it is producing equal half, identical left and identical right, then you can say that the organism is having a symmetry. What kind of symmetry it is further having, that is we will be going to discuss shortly. So this is the characteristic. So if I draw the diagram like this, let us suppose I am taking a cylindrical shape, that means a organism which is having a cylindrical shape. The center point will be here, somewhat here, okay? So you have to imagine a imaginary axis. So let us suppose that imaginary axis is passing through this center point of the body, correct? If the imaginary axis is passing along the length of the body, then we will call this imaginary axis as longitudinal axis. If it is passing perpendicular to the body, again from the center point only, then we will call it as a transverse axis. So if you divide with an imaginary plane along the line of axis and you obtain a equal identical left and identical right, then we can call it as that the organism is having a symmetry, correct? So either the organism is having a symmetry or either it will be a asymmetrical. The example of asymmetrical is definitely poriferans. Like amoeba, in the case of unicellular organism, we have seen that it is of irregular shape, so it is not having any kind of symmetry, it is asymmetrical. Similarly, poriferan members are also asymmetrical in nature. Of course, the exceptions are there, we will discuss in the porifera phylum only. So here we can see that we divide the organism in different plane, and then we see that in which plane it is producing identical left, identical right, okay? So let us see, if the organism is having a symmetry, the basic symmetry is the bilateral symmetry. Now how do we define bilateral symmetry? The one example is this butterfly, the other example is me itself, that means you all. So this is the other example of human. So if we have to define bilateral symmetry, that means the only condition is that in only one plane the division is possible. So if you try to divide me from this transverse axis which is passing through my center point of the body, you will not obtain identical left and right, identical half. But if you define along the longitudinal axis, if you divide like this in this plane, then you will obtain identical half. So if the organism is having a symmetry in which only one plane is creating identical half, then such kind of symmetry is called as bilateral symmetry. Moving it to the next part is the radial symmetry. So here we can see the example which I have taken. Again I will take the bottle, this cylindrical structure. So the cylindrical structure is more or less represented by this one. So the other radial symmetry example is, here you can see that if we divide in more than one plane, and at each division, at every plane we are obtaining identical half, then such kind of symmetry is called as radial symmetry. Radial means radius, along the radius. So if you took this example, if you can consider it as for a short movement as a cylindrical body, and if along the longitudinal axis I cut the organism body into two halves and I am obtaining equal identical left and right, and at each plane I am having identical half, then such kind of symmetry is called as radial symmetry. So here you can see and compare that asymmetrical is not having any kind of symmetry. This is the radial symmetry, that means along the radius, and this is the bilateral symmetry, this one. This is the example of sea anemone, again a cylindrical body example. Here we can see the diagrammatic representation also. I hope you are getting the picture of the radial symmetry and bilateral symmetry. So it's a, oh, I must hide it, the answer. You try to give the answer. This is the match the column, and these are some of the basic parameters written on one side along with the examples on the other side. I know that we have not studied the examples of the different phyla, But if we take a look at this, it's just an example of how the question can come in the form of match the following. So let us have a look. These are the parameters: organ level, cellular aggregation, tissue level, organ system. These are the different animals. Example, Pheretima means earthworm. It is having again organ system level of organization. Fasciola, that means you can see that here we can see the first point, that is Pheretima is having organ system level of organization. Second, Fasciola, that means liver fluid. Platyhelminthes, it is having organ level of organization. Now in this particular example, the student might get confused, as at the end of the chapter in the summary, organ system level of organization is also mentioned, but in the theoretical content, organ level of organization is mentioned. So if the choice has to be done in between the organ or organ system, we will definitely go for the organ level of organization, that is second for A. The third is a spongilla, freshwater sponge it is. Porifera example, cellular aggregate, it is the only exception in the cellular aggregation. That means poriferans are the only exception that are having the cellular aggregate organization. And the last one is the obelia, which is the example of the Cnidaria or Coelenterata, and it is having a tissue level of organization. So such questions can come, and many more variations can be made in such type of match the following questions. Right? So this was the end of this session. In the next session, we will discuss the next characteristics along with the different phylum. We will start from the Porifera and further. So I hope you like this. If you like this session, please subscribe our channel, and thank you for your time. Thank you very much.